Thank you so much for having me here. It's really an honor, and it's amazing to see so many people all together. So I'd like to talk a little bit about the work we've been doing over in Boston, and specifically really focusing on the genetics, and not just because genetics is where I've been focusing recently, but also because, frankly, a lot of us, and by us, I mean the researcher and physician community as well, find genetics a little bit scary, and the idea of risk factors a little bit unnerving. I can be told, for instance, that I have twice the risk of maybe being struck by lightning, and I know, for instance, that I've never actually known of anyone personally who's been struck by lightning, but I would still be quite freaked out. And much better advice for me would practically be, just having arrived from the States, look both ways before you cross the street, because it's really, I've already had a couple close calls. So what I would like to do is talk a little bit, actually, about genetics. What is the genome? Try to put that in the context of biology. I bet you many of you didn't realize you were signing up for a basic science talk. But then finally, talk a little bit about some of the new genetic testing that a number of you may already have results for or may be getting results for in the next couple of years. And again, trying to put that into a little bit of context. And the big thing I want people to understand is not to worry about this stuff. This is a very exciting and hopeful time. It's leading to a lot of new opportunities for therapeutics, for diagnostics, and that is the message I hope that people can leave here with today. So I'm from the Bing Center at the Dana-Farber Cancer Institute just across the pond in Boston. And as I mentioned, I study genetics, and the reason why we focus on this is that it's a very important part of, especially in cancer, of understanding how the cancer works. It can help us understand, create new targeted therapies, again, work in diagnostics, understand inheritance. There are, as many of you know, there are some families which have very concentrated clusters of WM. I'm not going to talk about that too much today, though I do want to take a moment and say, for the most part, this falls under other kinds of risk factors I just mentioned. You really shouldn't worry about it. If you're in a family like this, you don't need me to tell you. You already know. So really, again, this is not something one should worry about. So, understanding genetics. DNA itself is actually quite simple. It's just made up of 4 basic components. That's it. That's the entire thing. Lesson done. You can all go home. They pair with each other. The 4 different components are abbreviated A, T, C, and G. A is pairs with T, C with G, and it actually forms 2 complementary strands this way. You have a code going in one direction, and then a matching code that just perfectly matches that sort of ATCG pair going the other direction. And that's it. It's a code. You can read the code. And when we talk about genes, the secret here is all we're talking about is sections of the DNA that directly encode for how to build protein. So when someone talks about a gene, they're saying there's a chunk of DNA that they're interested in that makes a protein. And DNA being very important, of course, you want to protect it. So every time your cell wants to build a protein, you don't want to go to the master copy and start fiddling around with it unless if you have an option. So actually copies get made, and those copies are actually what get sent out and used as a template. And that's called RNA. So the other thing I want to talk about again is genetics. There is often a sense of genetics and predetermination, that somehow whatever your genetic makeup is dictates who you are. And for those who remember some of these studies that were coming out in the 80s, they were talking about identical twins, and they all use the same toothpaste and, date people with the same names and all this crazy stuff. And now that data's had a little bit of time to mature, actually, the real surprise is the fact that they don't actually behave very similarly, and they're actually quite different from each other, even identical twins. And there's a lot of reasons for this, and I would like to talk about that a little bit. But the big thing I want to make sure everyone comes away with is that the DNA is not a blueprint for who you are, and that who you are is much more than just the sum of your genes. So maybe the best analogy for this is actually maybe a computer program. Like DNA, it's a code. And as you can imagine, we could write out the entire human genome on a piece of paper, just like we could write out the code for some computer program on a piece of paper. There's nothing magical about it. It's just a code. With a computer program, we can even put it in a computer, but does that really, just seeing a computer sitting on a desktop, does that really explain what people do with computers? Did the person who designed the computer have to think of every possible use case that that computer would ever use in order to be able to program it correctly? No, of course not. That would be crazy. Every possible email that anyone could ever possibly write, you don't hard code that in. You allow these things to interact with their environment. So what is the natural environment, let's say, for a computer? Well, you can pull a researcher in, maybe a connection to the internet, and then, and only then, can you really start to understand what the meaning of this computer program is and what its possibilities are, because its entire point is to interact with its environment. And so DNA, like computer programs and other kinds of code, really cannot be understood outside the environment in which they exist. So what exactly is the environment of DNA? So DNA exists in the nucleus of a cell and is 3 billion base pairs long. It's kind of crazy. It's 102 centimeters in length. Now, the real mind-blowing part here is that this exists in every single cell in your body. 102 centimeters in every single cell. So, needless to say, this is a very, very, very tightly compact, very tightly controlled process. So, most of it actually isn't even available. And if you think about it, if your code is identical throughout your whole body, right? How is it that your skin behaves differently from your heart, from your liver? They can receive the same signal and they'll do slightly different things. And that's because they actually don't all see the same sections of DNA. It gets so tightly wound up that actually only small sections are available at any given time. They get unwound, they get used, and then they get rewound again. And depending on which sections are available, the cell will be able to sort of see different types of programs or different types of genes. And the basic idea here, in the basic model, at least when I was in school, and this has been modified, and we'll talk about it a bit, but you have DNA, and that stores all the information, right? Those are all the sort of hard-coded programs. You have RNA, and that's the copies of the genes that get made. So we can take a snapshot of all the messages that are coming off of the DNA that are being used to make protein, and that's RNA. And then protein. Protein is what actually gets formed. That's sort of the point of the process. And the protein does the work in the cell. So often what we're talking about functional stuff, we're often talking about protein. And the classical view is that the flow of information is always from DNA to RNA to protein. We now know that this actually isn't quite true. Biology is pretty much defined by all the exceptions to the rule. So there are in fact RNAs that regulate other RNAs. And as I just mentioned earlier, this winding and unwinding of DNA and also chemical modification of the DNA is something called epigenetics, which is actually orchestrated by the protein. And that can obviously affect what parts of the DNA can be observed at any given time. So for the first time, really, we are actually able to measure either all of this stuff or even a large chunk of it for an individual patient. And it's still pretty, it's still pretty complex, and it's not something we can just do as a routine screening visit. But we can really actually measure all 3 billion base pairs. We can really get a snapshot of all the RNA that's going on in a tumor cell. We can look at least a subset of the proteins. We can measure the epigenomic elements of some of the cells. And these, as you know, are all collectively referred to as omes. And now they have an ome for everything, it seems, including the bacteriome or the microbiome. Believe it or not, actually, even just at this conference, I had a really nice talk with someone who is mentioning that it's actually now well understood that that immune tolerance, the way that your immune cells are willing to tolerate or not tolerate certain things, is partially regulated by the bacteria in your gut. Who knew? And so there's a lot of interest, for instance, in with graft versus host disease in bone marrow transplant, actually working on feeding people good kinds of bacteria that might actually help them with that. So that's the kind of stuff that's now, thanks to research and things like this, are now coming up in clinical trial. And will hopefully be, assuming they work, will be available. And really what I want to stress with all these omes is that this is all stuff that's outside of the genetics, right? So the genetics is just one little tiny piece of this. And so when someone tells you you have a particular mutation, keep it in mind. This is one little piece of a much larger puzzle. And so how do we make sense of all this? All these different omes, 3 billion base pairs. People often imagine some either that there's some dark arts involved or that we actually sit down and read all this. But how much information are we really talking about? So when I go and I get a whole genome, I usually get it in pairs. I usually will completely sequence someone's tumor sample, so the Waldenstrom's lymphoplasmacytic cells, but then also some of the DNA that they were born with. So I can compare and see what's changed between the two copies. So comes actually as a series of text files, believe it or not. You can almost think of them as like Word documents, but it's about 600 gigabytes. Fair amount of data. So to put that in context using a local author, that's roughly 120,000 copies of the complete works of Shakespeare. Now, that's actually not entirely fair because these are actually highly compressed, and I personally don't know how to read highly compressed files. So if we were to uncompress everything, it would roughly fit in the size of a large library. 1.8 terabytes of information. So when someone, and we get this actually from doctors a lot, can you please send this to me in an attachment in an email? I don't really know what to say. And as a matter of fact, because it's so complex, we're still trying to learn how to interpret a lot of that. And I hope that in the future, as you start hearing about genetic results, whether you're reading about it in the paper, whether you're hearing it from your doctor, realizing that we're learning about this too, and that there's just as much that we don't know as we do know. Usually by the time that we start introducing it into the clinic, it's been very well characterized in a very particular context, but still keep in mind that there's a lot of unknowns, even now. This is actually, just for reference, a snapshot of an actual WM patient, and just looking at how we compress the information just so we can get the vaguest idea about what's going on. So what is the genomics of WM? Before we go there, I want to quickly talk about mutations. So what mutations are changes in the DNA. So changes in the code that have started to accumulate, and as they accumulate, I mentioned that genes encode for how to build proteins. So if you change the code, you also change the protein. And as you start to accumulate these, you can cause problems. Proteins that were supposed to be regulated one way are now regulated a different way. They now no longer have the same structure, and sometimes they gain completely new functions. And if you accumulate enough of these, sometimes one of the results can be that the cells start growing in an uncontrolled way. And when that happens, we call it cancer. Now, the best explanation for this I think I've ever seen was from Martin Krasinski at the BC Cancer Agency in Canada. And so I'm not even going to try to beat him, I'm actually just going to quote him shamelessly. but I would like you to help me out. So in the next couple slides, I'd like everyone to read along with me. And do you guys think you can do that? Can you do it out loud? All right. You guys ready? You guys are awesome. So what's the most common spelling mistake in WM? That is mid-88, which I think a number of you have heard about now. And what happens here is that there is a single piece of the protein that has been shifted. And what that causes is a conformational change. And normally what happens is mid-88 is an adapter molecule for something called the toll-like receptor system, which is just a fancy way of saying a system that's not particularly smart, but is very good at identifying common motifs in bacteria and viruses. So it's like an early warning system. And what's supposed to happen is when this early warning system goes off, two mid-88 molecules come together, and that starts the cascade. Once they're together, all these things start aggregating, including Iraq4, Iraq1, down to TRAF6, all the way down to something called NF-kappa B. And the problem is with this mutation is now they are able to aggregate on their own. They no longer need that signal, at least as far as we can tell. Or if they need that signal, they're now responding much stronger and much more consistently than they should. And we can actually see this in the cells. This is, here. Let's see if this laser pointer works. Yeah, all right. So you can see right here all these green dots. Those are actually little NF-kappa B molecules. They're all existing here in this blue nucleus. And that's where all the DNA is. So if they're in the nucleus, they're actually binding to the DNA and they're activating genes, and genes are being transcribed to RNA, and they're causing problems. So there's a lot of things we can do in the lab that unfortunately we can't do in patients because your body will just see it and break it down in little bits immediately. But one of those things is we can actually prevent mid-88 from dimerizing or coming together. And when we do that, it's amazing, we can kick it all out outside of the nucleus into the cytoplasm of the cell, where it can no longer interact with the DNA. And they actually start to die. So this is really exciting. And even at this meeting, we're now able to talk about new possibilities for therapies. There's one person over in BC, in Canada, who is currently working on trying to train your immune system to recognize mutant MYD88. Another person from our group, Sarah Berlage, who was talking about how we can possibly target these Iraq molecules to block the signaling. So there's a lot of really neat things that we can now talk about in terms of future therapy. They're not there yet, but now that we have an idea about what's going on, we can actually start talking about what we can do about it. So Leanne Zhu in our group has developed very sensitive techniques for detecting this. And this is a little strange. What we're measuring is time to detection. So the faster we detect it, the stronger the signal is. So the lower values are actually stronger and the higher values are actually weaker. It's really counterintuitive. And but what you can see is she went through and she tested all these different diseases. And really the only ones that were consistently positive were IgM-MGUS, which you just heard about from Dr. Bob Kyle, and Waldenstrom's. And one of the upshots of this actually is she's now adapted this test to work in the peripheral blood. Now we're not there yet, but one of the things we're hoping is that we can now maybe start doing this, using this for disease monitoring. I know how much you all love bone marrow biopsies. Hopefully maybe we can cut a few of those out now. We don't know, but this is the kind of thing that we can now talk about doing. The other interesting thing is that 3 MGUS people who had very, very strong mid-88 signal all transformed to WM shortly thereafter. So this is also the kind of thing that maybe we can start to use a little bit prognostically. Again, we'll have to see how this works out. most likely in the context of clinical trials. And it's not just us who have observed this. There's now been this large group of international researchers who have all gone back, used all sorts of different techniques, working on different tissues, who have gone and looked at the L265P MYD88 mutation in both WM and IgM-MGUS. And I have to say, it's been really kind of this amazing thing from starting in a small lab and making the discovery and now seeing this stuff reproduce all around the world. It's pretty cool. But I think the most exciting thing that's gone on is coming out from Guang Yang in our group. And he discovered that this molecule, BTK, binds to this complex. And it actually binds to this complex independent from these IRAK1/4 molecules. And it's actually able to bypass this whole, most of this graph really, and go straight towards signaling to NF-kappa B. And actually a lot of that signal is actually coming off of BTK, and this is all his work, really, really quite exciting. And the other great thing about this is we have a drug for that. So we actually got breakthrough status, largely thanks to the work of Guang and Steve, to look at using Ibrutinib in Waldenström's patients. And I think you'll probably be hearing a little bit about the clinical trial results at some point, but we got really amazing responses in IgM very quickly. I'm not going to talk about it too much here because I'm not a clinician and there'll be other opportunities, but I do want to say one quick thing, which is that, and this is interesting, the results in response were directly correlated to something called lymphocytosis. And this is the ability to get cells out of the bone marrow and into the peripheral blood circulating. And something we didn't entirely expect. But you can see people who got a partial response or a very good partial response had a lot more cells leave the bone marrow and enter the peripheral blood than those who didn't respond. And the reason I'm showing this to you is will become clear in a minute. So the other big discovery we had to do with CXCR4. And before I go there, I actually want to talk about something that has nothing to do with WM. And I want to repeat this again. Nothing to do with WM. I've actually never met or seen or heard of a WM patient who has this disease. It's called warts, hypogammaglobulinemia, infection, and myelocathexis. And it's caused by inherited mutations in CXCR4. And what happens is that CXCR4 is a homing molecule. It helps your immune system know where to go, and particularly it helps them home to the bone marrow. So by constitutively turning this on, people who have this disease, their immune cells go straight to the bone marrow, refuse to leave. They have a perfectly normal immune system, but just in their bone marrow. Good news is there's now actually inhibitors for this, and when you give it to them, their immune system goes back into the periphery and they have a normal functioning immune system, at least temporarily. Now, why am I mentioning this? We saw the exact same set of mutations, not in WM patients themselves, but in their tumor cells. The tumor cells had acquired very similar or identical sets of mutations, and this had led to constitutive activation of CXCR4. And all these mutations, as you can see, are clustered in just one small area of the protein, and it's something called the C-terminal tail. And the best way to think about it is, if you see where these arrows are here, this is the C-terminal tail, and all the binding and downstream signaling are happening in here. This is just regulatory. And the way this molecule gets turned off is quite literally the cell grabs a hold, you know, it sort of looks like a handle almost. The cell grabs a hold of that handle and yanks it inside the cell, and that turns it off. If you remove the handle, you have nothing to grab onto. You can't internalize the receptor anymore. It stays on the cell surface and just keeps on signaling. So that's what the problem is, and that's what we're observing in about 30% of WM patients. So Yankow and her group took a look at this, was able to actually show that when we introduced either the normal CXCR4, in lingo this is called wild type, don't ask, or the mutant CXCR4 into one of the two WM cell lines. In both cases, we were able to show after stimulating the molecule, there was a much greater level of internalization in the wild type or normal CXCR4 versus those cells who had been forced to express the mutant CXCR4. So again, what we're observing in WM is very similar to what we're observing in this germline syndrome called WHIM syndrome. And Yanko also went through and did some basic science work. Western blots are really just looking at protein, so the darker the band, the more protein there is there. And not gonna go into all the details, but ERK and AKT are really important signaling molecules. When we looked at baseline, you can see there's a lot more ERK and AKT for those who have the CXCR4 mutation. If you stimulate it, for everybody it goes up, but it goes up a lot for people with these WHIM-like mutations. If we introduce a CXCR4 inhibitor, it goes away. And this is again the point. Using information like this allows us to identify new possibilities for therapy. We know how to block CXCR4, and one of the things a lot of people are now looking at is how can we now translate this into clinical trials and use this therapeutically. Last thing I want to show is we also looked at Ibrutinib. And for the most part, adding Ibrutinib also shut down ERK and AKT, but if you look at those people who had the CXCR4 mutation, it didn't quite work. And if you remember, this is a molecule that helps the cells home to the bone marrow. And Ibrutinib is a drug that works partially by getting cells to leave the bone marrow and go into the peripheral blood. So we have a bit of a problem coming in here, and not surprisingly, when we looked at our trial results, and this is data that's going to be published soon, we actually saw much less of this lymphocytosis in the people who had a CXCR4 mutation. And this corresponded to response as well. People who had wild-type CXCR4 did a lot better. But, and again, I want to stress this, the people who had the WHIM-like mutations still had over 50% reduction in their IgM, and that's actually not too bad. So it's not something to worry about, but what this kind of data can tell us is what is leading to resistance, what we can do about it, and how we can introduce new therapeutic options to patients with WM. And in interest of time, I may skip over this, but we did see, you can track these things over time and see differences in hemoglobin and obviously IgM. And also when we go and we break down both MYD88 status and CXCR4 status, we're finally able to start to see differences between these different populations and start to make sense of some of the differences that we've always known existed in WM without, and just we never had an idea why or what was going on. And ultimately, we're hoping that this sort of information will help us develop new therapies, figure out which therapies we ought to be giving individual patients based on their genotype, and help people understand prognosis. The last thing I do want to say also is we did look some very initial data on overall survival, and you would think that having more mutations would be bad, and then actually in this case, having CXCR4 and MYD88 conferred absolutely no difference in overall survival at all. And again, the major emphasis here is please do not worry about these things. There was a little bit worse outcome for people who actually had neither mutation. And so this is a group that we're working on trying to understand quite a lot and trying to figure out what we can do to best help out these individuals. I think in the interest of time, I'm going to skip over copy number stuff, but really what I want to say is just that these are not the only two mutations, right? These are the two best characterized ones, the ones that we understand. There's a lot of other stuff going on, changes in the amount of DNA that people have, other kinds of mutations that are also quite interesting, such as ARID1A, CD79B, TP53, and we're working on trying to understand these. And again, in all that, as amazing as all this is, I want to go back to the very beginning and remember that all this needs to be put in context of epigenetics, heck, maybe even in terms of the microbiome, to everything it is about the individual person and who you are. And it's really only in the context of all this that we can really start to understand someone's disease. So don't feel at this point that these tests are somehow going to be defining. I mean, at some point we may learn more, but as of right now, I think that the big thing to take away from this is that we are now able to understand WM better. This means we can do better diagnostics. We may be able to reduce the number of bone marrows. And most importantly, we can bring in new therapies, and therapies that are maybe better targeted to the individual, and hopefully someday maybe a cure. So thank you very much. I'd like to thank everyone involved in the Ibrutinib trial, since I use their data quite a bit here. That would be Steve Trion, Rajana Advani, Leah Palumba, and Christina Tripsis. And then also everyone at the Bing Center, along with the Peter Hon Bing Foundation, the International Waldenstrom's Macroglobulin Anemia Foundation, who's helped out enormously in this research, the Coyote Fund for WM, the Bailey Family Fund, and the D'Amato Family Fund for Genomic Discovery. Thank you very much, and I'd be happy to talk to anyone later on.